Since I have your attention, I may as well just start, am I right? Yes. Welcome to Hunger Games. My name is Steve Cole. I'm just some guy, but I happen to have what I think is one of the coolest jobs in town. I get to run the Economic Development Foundation. What does that even mean? We're a nonprofit and we provide economic development services to the town of Plymouth and the region. Basically, we focus on small business startups. We try to encourage entrepreneurship. We help the town and surrounding towns achieve their economic development goals. It's about 25 years old, and some of the things that we've done to date are help create the Children's Business Fair. I don't think anybody in this room would have participated in that, but we focus on fifth graders, mostly because, well, when you look under the bed and you know there's no monster anymore, it's not scary to do as an adult. I mean, the only thing I can't do now that I could do in fifth grade is skateboard and long division. So one of the things that we try to focus on is instilling in students the skills that they need to not just run a business, but later on in life when it comes time to make the decision, do I go to college? Do I get a job? Do I join the clergy? Do I join the service? No one ever told me when I was graduating high school that you can actually make your own job. It's not easier. Trust me when I say this, it is not easier, but it's another alternative to you. So one of the reasons that we're here today is to talk about the Blue Economy Conference, right? This is day 2 of the conference. In fact, this is our 4th year, and each year this conference has grown, dare I say, it's doubled in size. The first year we did this 4 years ago, we had 30 kids in the room and no young women. And year 2, we had 53 students in the room with 3 young women. Last year, we had 103 kids in the room, 24 young women. And we did an informal count today. Sarah, what are we up to, about 50? About 50 young women in the room. I think that deserves a round of applause from you all. So during the pandemic, my board of directors, there's 13 of them. I like to joke Jesus did so much more with just 12. But over the pandemic, we were not dormant. We actually worked really hard to try and understand what this community needs. We wrote a grant to the US EDA, kind of a masochistic federal organization that mostly invests money in chronically distressed communities. Plymouth is not that, but we are undergoing a decommissioning. The power plant, you all know, has undergone a decommissioning. Really robust conversation right now about the cost for energy and whether or not we should reactivate that power plant. The purpose for the UMass, the EDA grant was essentially to study entrepreneurship in Plymouth. How do we recover some of the jobs that we lost? What are our strengths? I believe in asset-based economic development. Plymouth has 37 miles of coastline. There's a lot of access points to that coastline. We have a tranquil bay. We also have access to open ocean through the Cape Cod Canal. So if you're looking at doing something in robotics, and you'll see a lot of representatives here who work in that particular field, Plymouth is really a good place for you to consider starting up your company. There's a company here in Plymouth called Green Sea IQ. I encourage everyone to get familiar with that company. They are here today. I saw Don earlier. Ben Kinnaman was our keynote speaker, our first conference. And one of the things that stood out to me was he grew up in Vermont, and that's where the company was started. But he learned that we know more about the surface of the moon, the surface of the moon than we do the bottom of the ocean. And that helped inspire him for creating his company. There's 3 robotics companies that operate right now in Plymouth. Take a look at what those things are. It's phenomenal work. There's plenty of room to grow in that field, guys. That's one reason why we're here. So the US EDA grant that I mentioned earlier, we hired UMass Donahue Institute. They studied for us who we have in our ecosystem as an entrepreneur. What are they working on? What resources are they using, and what resources do they need to scale up? So my foundation, in partnership with the town, we're ready to respond to you. When you're ready to start your company or look for that job, that's what we're here for. So your only job today is to find out what you might want to do next in your life. That's really it. We're just going to expose you to a number of options for you, okay? Myself, I'm here as a resource to you. I saw Michelle earlier. Michelle, where are you? Are you in the room? There she is in the back. So if you have any questions today about the conference, what to do next, what you have to do, find me or Michelle. She's in the back wearing that, what is that, mustard colored jacket? Mustard? Mustard. Thanks, Michelle. So there are 22 resource tables. We have mock interviews. Some of you have already signed up for those. We also have a student survey. You're encouraged, please, to fill out the student survey. This gives us really important feedback on how we can make these conferences better for you. If you've attended a conference in the past, if you've been here for the last 3 years, we have a special gift for you. So find me later on. I'm going to get your name on a list. We have something upstairs that we can give to you. For interviewers, we have a couple of options for you as a free gift too. We have this absurd little plushie that your dog will absolutely love, right? This is our mascot. You see the tail there? This was informed by Henry David Thoreau, who wrote in his journal about a sea serpent who lived in Plymouth. Real eyewitnesses were in that account too. So we adopted that as our logo, our mascot, because if there's anything I've learned about working with entrepreneurs over the last 20 or so years, there be dragons and they know exactly where they are. They don't always know how to overcome that dragon, that serpent, but they always slay that monster every single time. That's why we have such a high rate of success in Plymouth in entrepreneurship. By the way, 15% of the jobs in Plymouth are held by home occupation, which means our commercial environment is so built out, people are actually running businesses out of their homes. So there's a deep, innovative, and entrepreneurial spirit throughout the South Shore, and I know that you're all a part of that community. So if you participate in an interview, you have a choice between this free gift or this free gift. And if you or your mom or anything like me, I like to drink wine in the morning very discreetly, and this is one way to do it. Today's event is sponsored by the Education Foundation. I represent the Economic Development Foundation. Later on today, hopefully you'll hear from Sean Costa, who is the president of the Education Foundation. They put up the money to make this event possible. So I know I've already asked you for one round of applause. If I could ask you for a second round of applause for the Education Foundation. And I think I have successfully filibustered enough. Oh, there's one more thing. In your bags, you have here these passports. These are also sponsored by the Education Foundation. These passports are intended to give you access to the very people you're going to need to talk to when you're ready for an internship or to apply for a job. Every one of the resource tables have one of these stamps here. So we want you to receive the stamp to show that you were at the table. I want you to record the name and the organization that you are speaking to, and then just any general notes about that organization that you find interesting. You're going to put this in your desk drawer, and you're going to forget about this for a couple of years, perhaps. We made it bright green so you know where to find it when you're looking for it. When you're ready for that internship or that job, this will be your first point of contact. You're encouraged to reach out to some of these people that you meet here today to help begin your career pathway. And with that, I'd like to introduce Kristen Osborne, who's a Mass Maritime professor in marine sciences, safety, and environmental protection. Kristen has a PhD in marine science and technology, and she has a passion for sea squirts. In fact, I know, I heard you all had the same thought I had, but it's a sea squirt, and she's going to tell you all about it. And she leads these invasive species treasure hunts so that you can see not just the sea squirts, but other types of invasive species that exist in our area. Ladies and gentlemen, can we welcome Kristen Osborne to the stage?
Good morning, everybody. How we doing? How are we really doing? Woo! All right, I'm very excited to be here because this is my first time being at the Blue Futures Conference, and I'm even more excited because one of my students is going to be presenting today. So I'm honored to introduce our first presenter, Michaela Accord. She's one of the best and the brightest at MMA. She's a senior undergraduate at studying the Marine Science, Safety, and Environmental Protection major, and I know that's a mouthful. You may have heard of MSSEP, so that's where we're from. She's also getting a concentration in marine biology. She's worked at Woods Hole Oceanographic Institution, or WHOI, which I'm sure you're familiar with at this point. They are the leading independent nonprofit organization dedicated to ocean research, exploration, and education. Michaela started out at Cape Cod Community College, and then she interned at WHOI and has been using those connections ever since to continue her involvement there. Today she's going to be presenting her independent research project that she worked on last summer, and spoiler alert, sea squirts. So you're going to learn all about them from her. I'm going to pass the mic for that. And so her presentation is entitled Low Salinity Tolerance and Recovery of the Invasive Tunicate, aka sea squirt, Botuloides violaceus. And she's exploring how marine invertebrates are being affected by climate change. This research is adding to that growing body of knowledge that we're using to track and also to mitigate this problem, because it is one of the major issues that we face today in terms of threats for global biodiversity. And those impacts are intensifying with this changing climate. She's offering valuable insight into opportunities for folks like you who are passionate and interested in pursuing a career in science. So please make sure to visit her and our other students that have come from the Academy today at our resource table. Please join me in welcoming Michaela Accord.
Good morning, everybody. I am so excited to be here and so excited to share the research that I have done at Woods Hole Oceanographic Institution. So a little background about Woods Hole Oceanographic Institution. It's also referred to as WHOI. It is local. It's in Woods Hole, Massachusetts, so the very, very bottom of Falmouth on Cape Cod. It was founded in 1930. And like Professor Osborne said, it's the foremost independent nonprofit organization dedicated to ocean research, exploration, and education. It is such an amazing institution to be at, and you guys are all lucky because you live so close to such an important organization that's making some of the most leading discoveries and movements in ocean science. The goal of WHOI is to further the understanding of the ocean and its importance for the entirety of the Earth through science, education, and engineering. So everything that you are learning about today is directly applicable to what WHOI does, and if you're interested in going to WHOI for work or internships, then that's such a wonderful place for you to connect with. So my position there, I started out in a blue economy internship through Cape Cod Community College, and then I transferred to the CC crew for community college students, and then from there I was able to have my own individual position as a guest student researcher. So through that, I performed individual and independent research in the Teapot lab, so I worked in the biology department mainly. I also worked in the geochemistry lab before that. And with my project, I studied the salinity tolerance and physiological influences of the invasive tunicate species, or sea squirt, Botulides violaceus. And through my work there, I was also able to attend lots of lectures. There, not only is there science and research going on, but everybody loves to share what they're learning. And so I was able to talk to a lot of people and make those connections. And then I was also able to compile a poster and present it. I talked in January at the Society of the Integrative and Comparative Biology's National Conference in Atlanta, Georgia, and I cannot stop talking about my research since then. So a little introduction into the species and what they're doing, where they're from. So Botulides violaceus, I know it's a mouthful. I apologize for that. There is no common name for it, but just bear with me with the pronunciation. But it's an invasive colonial tunicate, so invasive, it's not native to most of the places that it's in, and it's colonial, so little zooids, they look like this under a microscope. They colonize into these big masses, and that's how they're very distinguishable. They're native to the Northwest Pacific, so in the map, that's the blue area, and they're now invasive into the Northeast Pacific, Northwest Atlantic, and regions of the Northeast Atlantic. So if you go out onto a dock by your house or your school, and you look over the side and you see a big, disgusting-looking orange glob, it might be Botulides violaceus. They can travel and invade through settling on the sides of boats and other marine settlement areas, and then they can also be in the ballast water of ships. So they're very invasive, and they can travel through the shipping industry, which is applicable to the blue economy. And they have the ability to grow very rapidly and reproduce rapidly, which makes them a very strong competitor for space and food. So they threaten native species such as other tunicates and shellfish. So they can actually be very detrimental towards the aquaculture that is local to Massachusetts. So with climate change, it is increasing the frequency and intensity of storms, which means that there's a lot more, say, like hurricanes or huge rainstorms. And with every storm, that's just dumping a lot of fresh water into the ocean, which changes the salinity or the amount of salt that's in the ocean. And that can have an impact on all species who might have specific salinity tolerances that they can handle. And the average salinity for the ocean is 35 parts per thousand. And I wanted to see what the salinity tolerance of this specific species is. This is what an adult colony looks like. It actually grew over something else. So the matting, the orange matting, that's the species itself. That's Botulitis violaceus, and it looks like it grows in chains. So sometimes it is referred to as the chain tunicate. So the purpose of my research was to understand how botulitis responds to and recovers from periods of exposure to low salinity, so more fresh water. And I wanted to see how this might influence their potential future to spread, where they could invade in the future, and how they might be affected by continuing climate change. So the objectives of my actual research, the three things that I wanted to find out, was the median lethal salinity, or LS50. So that's where, that's when you look at one salinity point, and if 50% of them die off at that point, then that's the median lethal salinity. And I wanted to see what the median lethal salinity was for samples that I took right off the dock in Woods Hole, and I wanted to compare it to previous data that we had from Sitka, Alaska. I wanted to see if the botulitis could also recover after having no recognizable heartbeat. So if they're in fresh water for too long, then we can't see their heartbeat under a microscope anymore. So I wanted to see if we move them back to normal or ambient salt water, if that would change the heartbeat. And then I also wanted to see if these exposures to the low salinities would change the way that they grow and their overall health after that. So how I designed it, it was pretty simple, a lot of cheap equipment. A lot of people think of science and multi-million-dollar projects, which that is true, but you can also do science with very limited materials and cheap materials, which I think is also pretty amazing. So I took these just plastic well boxes and I lined them up with microscope slides so that hopefully when I put in adult colonies of botulitis, they would reproduce and they would release. They almost look like little sperm cells, but they're orange, and they are the larvae, and then they swim around and they settle on the microscope slides. And then hopefully when I take it out, I can see it well underneath the microscope. So I just left them in normal ambient seawater, which is 35 parts per thousand, and I put a light above them, and I let them sit for 24 hours to promote this larval sediment onto the glass slides. After that, I took the microscope slides out, checked to make sure they had a heartbeat and I could see it well, and then I distributed them amongst several salinity treatment tanks. I did salinity levels from 10 to 35 parts per thousand, so 35 again is average, and I was looking at the lower end. So I looked at 10, 12.5, 15, 17.5, 20, 25, and 35. And then I let them sit in those salinity treatment tanks for 20 hours before I pulled them out and then looked at their heartbeats underneath the microscope. So again, after 20 hours, then I would check for heartbeats, mark to see if they had one or if they didn't have one, and then I would return them back to normal seawater to see how they might grow and if their heartbeat could potentially change. And then after that, I just left them in regular seawater and I looked at them for heartbeats and to see how many more zooids they could grow. So an individual, like fresh, almost like baby tunicate is called a zooid. And that's just like on the right image, that's just one of the circles. And then when they continue to grow, then they just create new ones. So there's two in that one. And I checked them at 24, 48, 72, 96, 192, 288, and 366 hours. So what I actually found, I know it can be hard and intimidating to read graphs sometimes. I still feel the same way when I read research papers, but I'll try my best to explain it. But this just shows the survival rate against the salinity treatment. So I actually have Massachusetts and Alaska data on this, even though I was only responsible for the Massachusetts data. So this shows the LS50, so the median lethal salinity, where 50% of the samples died off at, depending on what salinity treatment they were in. And so the dark orange line is Massachusetts, and I used statistical analysis to figure out that 50% of them died off at 15.07 parts per thousand. And the lighter orange, almost yellowish color line, that is Alaska, and it was actually very similar at 15.11 parts per thousand. And then all of the individual dots are just individual data points as well. So some of them, as you can see the bottom line, or the bottom two dark dots at 25 and 35, those are just random samples who decided to die on me. There could be any reason behind that, but I just wanted to display that as well, that not all of them survived, even though they were in conditions that they should have survived in. This shows the oozoid recovery, so how their heartbeats looked after salinity treatment. So what was surprising that we haven't seen before is the botuloides can recover after having no recognizable heartbeat when they were in a really low salinity. We haven't seen this before, and I actually discovered it by accident. But I had a slide with several oozoids on it, and one of them died, but the rest of them survived, so I just put it back in ambient seawater, and then I checked all of them like the next day, and they were all alive. And I was like, is this a zombie? Did it come back to life? Like, what's happening? And I talked to my boss at Hui, and she was also very surprised. But that completely changed the way that we were running the project to make sure that we weren't just putting the living oozoids back into ambient seawater. We wanted to put everybody and then change, hopefully that would influence our data set to really get a holistic view of what's actually happening with these tunicates. So this graph, it shows 3 different proportion graphs, and the top one is 10 parts per thousand, so that's the freshest water that I had, and then I had 12.5 and 15 parts per thousand. The x-axis shows time in hours, so directly after I put it back in ambient seawater, which is 0 hours, and then all the way up to 360 hours at the end. The orange color means that they have a visible heartbeat, so that's like the proportion or percentage of them that do have a heartbeat that was visible to us. And then the gray area shows having no visible heartbeat, which that doesn't mean they're dead. It just means that they might be in a process where they're stopping their heartbeat to try to survive in the future. So even though they can not come back to life because they're not dead, but even though they can recover and regain their heartbeat, it is more common. The higher the salinity, the more likely it is for their heartbeat to recover. So in the 15 parts per thousand group, you can see there's a lot more orange, so a lot more of them were able to recover. Whereas in the 10 parts per thousand, there were some that were able to recover, but it was less likely. This was very exciting for me to kind of not have like a scientific discovery, but just to see something that we weren't expected at all. And it's something that I personally can contribute to the field of science that isn't there before. So we're filling in the gaps that people didn't even realize were there. So then another result that we found was that the botuloides vivacious growth, it was reduced after the exposure to low salinity. This was kind of expected, but it was nice to just have this confirmed. So this graph shows the number of zooids. So do you remember the graph where I showed the 2 orange, like almost bulbs next to each other? That was 2 zooids, and they would continue to reproduce and grow. So sometimes I would look at them and there would be like 7 by the end of the experiment at 360 hours. So I tracked the number of zooids, and that just showed how productive they were and how well they were doing, how well they were able to reproduce. And I checked this at 192, 288, and 366 hours. And I compared it all by what salinity treatment they were exposed to. So the final average zooid growth, because I averaged all of them, after 360 hours shows that low salinity exposure actually negatively influences the number of zooids that were there at the end of it. So with the graph on the side, the highest line, so the highest average number of zooids was 35 parts per thousand. This is just normal seawater. Great, if the sea's normal, then they're gonna produce normally. That's wonderful to know. But then we also see that if it was really fresh water, then the average number of zooids was extremely low. And this it kind of shows in that area how low salinity does really influence how they are able to be productive. So this was very interesting because botuloides does have salinity constraints, and most species do have salinity restraints, temperature restraints, even pH restraints that could impact where they could potentially invade in the future and where they are right now. And this is directly related to climate change because as humans, we are influencing the climate at a higher rate than ever before, and because the climate is changing faster than it has, it is up to us to decide how we proceed in the future. So this is very important that you guys are here because you can learn a lot more about how you can have a role in maybe making a positive influence on how the climate is changing in the future. Something else that we also discovered, again, is that zooids were able to temporarily halt their physiological processes when they were stressed. This was very exciting. We haven't seen this before, but I hope to do future work in researching more into this, or you guys could do that. The changes in salinity from the storm events can also have a prolonged effect on the growth of these botuloides species. So even though they're able to survive a low salinity exposure in the moment, that doesn't mean that they can just recover and be as good as everybody else. It's like when you sprain your ankle at track practice or something. Sure, yeah, you can run after, but maybe you're going to be slower. You're not performing as well. It's the same principle towards these tunicates. So in the future, what I want to look at is how botuloides oozoids respond to and recover from longer periods of exposure to salinity. So I just looked at a quick 20-hour exposure. That's pretty typical for a big storm. But let's just say there's a hurricane going through, or like this past week where it just kept raining. It did not stop. I know I was tired of it. You guys are probably tired of it, and the oozoids are probably tired of it. So it would be interesting just to see if the results would vary based off of how long they were exposed to these low salinities. I would also want to see how the colonies that are found in brackish water would respond, because I took my samples right out by the ocean. So it was just normal seawater, but brackish water is that kind of combination of salt water from the ocean and fresh water from land combining together to create a new mixture. So the salinity varies a lot in those areas. And I want to see if those routine salinity fluctuations actually influence their resilience to low salinity conditions. I also want to investigate the potential for genetic variation and transgenerational plasticity to influence salinity tolerance. That's a mouthful, I know it is, but just to see if there's any genetic reason why some of them could react differently to these salinity exposures, and maybe some of them are better off to fight the salinity exposures than other ones. And again, I want to stress how you guys all can help, because coming into science can be extremely challenging and intimidating. I know that when I was in high school, when I was in your shoes, the one thing that I did not want to study in college was science. It scared me, and I felt like I wasn't smart enough, or I didn't have as many opportunities as other people, and it would be better if I just studied something easier and more comfortable. But taking a marine biology class in high school really opened up my eyes to it and taught me that even though it's not comfortable, it's still something worth pursuing, because the amount of gratitude that I feel when I can talk to people, and even though I struggle through classes, but they're willing to help me and I'm willing to pursue through it, it's really rewarding. And hopefully you can make a lot of connections here today to further your future in science, because we cannot continue to make such great findings and such great influences on the world of science if we don't have great young minds like you guys. There are also lots of opportunities. I'll talk about this at my table later, but if you're interested in Mass Maritime and MSSEP, I would love to talk to you. But there's also a lot of other wonderful resource tables for you guys as well. I just wanted to thank everybody at Woods Hole Oceanographic Institution and the Blue Future Conference for the opportunity to talk to you guys. I am truly humbled to be up here. I still can't believe it. I'm a senior in college, but I feel like I was just in your shoes. So just remember that anything is possible if you put your mind to it, and you can truly have a large impact on science and other people. If you want to talk to me later, I also am joined with two seniors from Mass Maritime, Dan Kowalski and Aiden Webb. They are bright and brilliant, and I'm so lucky to have them here. So if you would love to talk to us, then I would love to talk to you as well. So please find us at our resource table. Thank you.
Was Michaela not phenomenal and inspiring? Michaela, where are you from? Sandwich, just over the bridge, huh? Local enough. One of the coolest things that I've learned about doing these conferences over the last few years is that Ashley Stolba, who is the state's Secretary of Economic Development, Katie Acheson, who's here today, from the Cape Cod Chamber, and myself, we all grew up on Cape Cod also. So I find it really compelling and interesting that someone from what I call Peninsular Plymouth is actually leading the conversation across the state for blue economy.